who did his PhD in Leibniz in Institute in Plant Genomics, Genetics and Crop Plant Research in IPK in Germany, and awarded PhD degree in Martin Luther University in Wittenberg. Then he worked at the Max-Planck Institute for Plant Breeding Research at the University of Zurich as a postdoctoral researcher. Since May 2017, he became an independent group leader at the Institute of Crop Science at the Chinese Academy of Agricultural Science in Beijing. And he and his team are working on barley germplasm research and their use in basic and applied research. Today, Dr. Yang will invite one of his PhD students to present the second part of the presentation, who is working with him on barley genetic resources. His experimental skills include population diversity analysis and map-based cloning. So the seminar that Dr. Yang is going to present today is HTX mutagenesis population for barley gene discovery. So now let's welcome Dr. Yang and start his presentation. Thank you.
Thanks, Sebastian, for the kind introduction. And also, I'm very glad to have the opportunity to show the work in my lab. So in this International Barley Hub. So today I would like to introduce some work in my group. So the HTX is so basically it's a Chinese languages. So in Chinese it's HTX. So then you can see what you can see. So this is the phenotype of these germplasms in the field. So when it becomes maturities, the whole plants will become black, including the spikes, stems, and leaves. So before I start my talk, I would like to briefly introduce my groups. So my group is Barley Genetic Resources from the Chinese Academy of Agricultural Science. So my group is working mostly in the barley germplasms innovation and utilization. So we hope our work would be helpful for the barley breeding and basic research. Basically, so in my group, our group, so we have three works. So including the germplasm evaluation, innovation, and also the gene characterizations. So this is my group pictures. If you are not familiar with our task, so you can consider the work performed by the Haruhiro Sato from the Okayama from the Okayama universities. So our work would be quite seminars. So here, so where we are, so we are basically located at Beijing, the capital of China. So this is our working building. So it's a National Crop Gene Bank of China. So our group is just at the fourth floor. So this would be, my office would be around here. So what you can see here, this is a view of inside of these buildings. So this is the architecture of wheat, and this is a letter. So basically, it's a seed means seeds in Chinese. So so now come back to my talk. So in this talk, I would like to introduce three parts. So including introduce the HTX mutagenesis preparation, and also give some examples to show the forward and reverse genetic studies. As you might familiar with the barley mutants, so the barley mutagenesis studies have been started nearly 100 years. So nowadays there are a large number of barley mutants and which is reserved in a lot gene bank. So as you can see here, this table, which is published by the UDA. So you can see there are a lot of mutants and mutant alleles. So regarding to the morphologic phenotype traits and also the pathogen resistance. So the mutagenesis not only used for the basic research, but also very important approach to in the breedings. I would like to introduce two examples. For first example would be the golden promise. So this is a very well-known British cultivars, which is derived from the radiation treatment of the cultivars method. As you may familiar with, this is a very nice cultivars to use for whiskey production, and maybe even more well known because it's a very famous cultivars used for barley transformations. So another example would be from a Chinese varieties Yanfu Aizao 3. So this is semi-dwarf malting barley and derived from a Japanese cultivars Zao 3 or in Cantoniju 3. So this is the biggest Chinese varieties growing in the Chinese barley field. So in another hand, so the barley mutants are also used for genetic studies. So once we observe some phenotypic variation, so we will so the mutants would be a very good tool to for the genetic study to identify the molecular basis behind. So for instance, there is the one gene VRS2. So this gene modulates the later spike fertilities, and so this is identified from the mutant. So once we identify the candidate genes for the mutants, we are also able to use the mutant to validate the gene functions. For instance, is another story, which is well-known genes MAT-C. So this is you can see here if there is a mutation for the candidate genes HVCN. So you are these to the early mature flowering. So this is also a very famous stories and also very important genes in the European barley breeding. 20 years ago, Robbie's group established the first killing preparation. So because once we have the candidate genes, or we need a tool to test the gene functions. So how to identify the mutation sites at a particular site? We need a approach. So 20 years ago, Robbie's group reported the first killing platforms from the British cultivar optic, and after that, you can see here there are a lot of several team preparation have been published in several countries. However, when I start my work in China in 2017, there is not such a resources in Chinese barley research communities. So because we know this is a very important resource for the basic research, so we would like to generate these resources for the future studies in China. At that moment, we decided to use one barley landraces, which we consider is a Chinese barley landraces, HTX. So basically, it's HTX in Chinese. As you can see here, this is a very special genotype because when it becomes maturities, so the whole plant becomes black. Also, we are also interested in the virus resistance. So because this genotype is susceptible to virus infections, so that would be allowed to identify the mutant show the virus resistance. However, when we analyzed the population, the genetic the PCA structures, we realized HTX is not a traditional Chinese landraces with the Eastern genetic background, but basically this is a mixture between the European and Ethiopia's genotypes. We also tested this genotype because for some green qualities, as you can see here, this is also a little bit special because they show very high protein content, but slightly lower starch content. So that means this would be a good material for say to work on the green qualities related traits in the future. So when we started this work, we first tested the EMS, the mutagenesis solution to see to find the best dosage for the treatment. We tried several concentrations, and we realized so once the concentration is more than 40 millimolar, so this would be only less than 10% plants would be fertile. So as you can see here, so and finally we decided to use two concentration. So one would be the 22, another one would 32. So we combined both conducted treatment separately and to produce the mutagenesis preparations. And in addition, we also trying to perform the second treatment by the EMS mutagenesis treatments. So, and we get more than 1,000 lines. Also, we included the material from the test experiment, as I mentioned before. So finally, we get stocks with more than 10,000 lines of variables. So with the seeds available. So when we have this mutagenesis populations, we try to generate attaining plant forms. So as you can see here, we selected, because barley has a very big genomes, so we believe we are not able to sequence all of them by whole genome sequencing. So in order to consider the budget, so we decided to use this amplicon-seq approach. So we use 3-dimensional pruning strategies, as you can see here. So we pulled 144 samples in one super pools. So that means we have this is 3 dimensions. This is 1 dimension, another dimension, and this is 3rd dimensions. So we have we generated 3 sub-panels. So each panel contained 1,728 samples. So we have 2 versions. The first version contained more than 5,000 lines. All of them are M2 plants, because the M2 plants have some problems in the seed germination. So we also extended, generated a second version of the teeming plant forms. So basically collected its offsprings. So at the M3 and M4 generations. So we also tested analyzed the mutation frequencies in this population. As you can see here, so there are several published mutagenesis populations. We also analyzed HTX with other published populations. So basically, with our result, so the HTX population the mutation frequency would be around 250 kb per. So one mutations per 250 kb. So that means we believe this is one of the best mutagenesis populations that would be used for support our future study. In addition, we also in collaboration with Martin Marshall and Neil Stein, so we generated the reference genomes for this genotype. So the first version of the genotype is rather fragmented. So and later we use another use the PacBio Hi-Fi plus ONT and Hi-C. We generate a second version of the genotype of the reference genomes. So the new version of reference genomes would be the quality is comparable to the Merck's V3. So the N50 is around 80 megabits, because we realize Merck's version 3 is a very wonderful reference. However, we also realize some cases, some genes may be absent in its reference genomes. So therefore we decide also, we believe this reference genomes would be helpful to identify some genes that are absent in the Merck's reference genomes. So now I would like to move to the second part of my talk. So in another hand, we also identified a large number of mutants that show the phenotypic alterations in the populations. As you may see here, some mutants, some lines show the upstream leaves here. Also, some so they show the complete yellow plants throughout its life cycle. So this becomes yellow since at the seedling stage until the harvest. So also some lines show the disease mimic leaves, or some lines show the complete show Wackley's phenotypes. We also find some phenotypic variation at the spike. So for instance, this is well-known locus 6-lord spikes, which is controlled by this VRS1 genes, and also some dense spike, sealed spikes, and was the muffled florent organs. So we identified, we didn't carefully analyze the number of lines show the phenotypic alteration, but briefly we collected more than 1,200 mutants. However, so the lines show the phenotypic variation, the number should be much high, more than these numbers. So once we identified the mutants, we would like to clone these genes by by the forward genetic approach. For instance, this is the first story I mentioned before. So this mutant shows the chlorotic phenotype. So we use the in collaboration with Martin Masher. So we use the booked segregates analysis and finally to identify these genes. So this gene is published, reported before. But this, at least the principle we used is work. We also, for the second example, would be another qualitative traits. So we refer to muffled fluorescent organs. So basically, this is the MFO phenotypes. So if you compare it with the wild type, so this mutant shows the multiple arms phenotype. So all the fluorescent organs becomes lemma-like phenotype. So for instance, the 3 stamen converted into the lemma-like structures. So we clone these genes, use the mutant map approach. So another story would be the multi-ovaries phenotypes. So this mutant show 4 ovaries. So all these 3 stamens converted into the ovaries phenotypes. So this gene is encoded by matchbox genes. We also identified some genes, but were already published. So, for instance, so this mutant shows the albino lemma1. So, as you can see here, compared to wild type, so the lemmas becomes yellow or black, white or black, yellow, and also there is something different at the stems and the nodes. You can see here, but these genes are already published by the Gatersleben group. So in addition to the qualitative traits, we also try with this resource, we also would be helpful to identify the quantitative traits. So for instance, this is story we refer to this thrust spike mutant SS2. If you look at this phenotype in the nature populations, because this is determines the length of the peduncles, the interlude, the first interlude just below the spike. So this were definitely considered as a quantitative traits. But however, in this mutant population, we know so the phenotype in most of them should be caused by the single mutations event. So once we identified these mutants, so we found two independent mutants show the allelic variants. So we also use the mutamap approach. So you can see here is mutants. So there is no difference at the other internodes, but if you consider the length of peduncle, so it's dramatically reduced compared to wild type. So here, so why we focus on, we will notice these mutants, because in the west part of China, so there is some very, some Chinese varieties, so show this thrust spike phenotype. So usually, if for instance, this is one cultivar Quinton 15, and its sister cultivar Quinton 14. If you compare two cultivars, so the cultivars with thrust spikes will show more than 30% spike numbers per areas. So this would be a very important trait to increase the yield productions for barley varieties, and these traits are highly selected in these areas. But of course, the genes we clone is not directly this gene, but I would say this phenotype would be very important. In addition to the vegetative traits, we also pay attention to the calyx-related traits. As you can see here, this is HTX. So this is seeds of HTX, and we also observe a large number of mutants. So the morphology, show the calyx related variations. So some show the enlarged green length, liquid greens. Basically, this is different with NUD locus. And also some mutants show the black calyx, but with yellow lemma and palea. So some mutants show small greens, also were the endosperse abortion phenotype. We use the same strategies to identify the genes, but because we are not really work on this field, but just a test of example concept, we work on the two mutants. So we are able to run to identify the candidate genes very quickly. So for instance, this mutant, we only observe one candidate genes. For another mutant, there are 6 candidate genes available. So that would be a few candidates for the following gene validations. So now I will move to the third part of my talk. So as I mentioned before, so the mutagenesis population was a very important resource for reverse genetic studies. Once we identify the genes, we would like to test it, how this genes works. So if we have the mutants for the particular genes, that would be a very important resource for gene functional analysis. Just one example, so the bright one. So this is a very famous semi-dwarf mutant Wuzu, which is first reported from Japan. So this Wuzu phenotype would be this allele is able to modulate plant height, spike length, and also the spike density and the spike length. However, this mutant is able to reduce plant height, but also reduce the grain size and solid grain weight. So this locus, so this This Wuzu alleles is a so super strong phenotype, and this is not good for the barley breeding. So, and for instance, right now in Chinese varieties, in modern varieties, the Wuzu alleles has been discarded in the breedings. So with the resources, with the teaching platforms, we are also able to identify some alleles at this BRI1 locus, but show the moderate phenotype. So you can see here, this is just two examples which show the slightly reduction on the plant height, but not observe significant difference on the spike length and the green, southern green weight. So another example, as I mentioned before, so this was for the MFO mutant. So we also identified additional mutants. So this mutant would be able to convert the candidate genes which identified in the forward genetics. So the third would be the SS2 I mentioned before. So we identified a number of around 9 independent mutants at the gene coding sequence. So all the mutants show the short pedancal phenotype. But however, so different alleles might show the different dosage on the length of the pedancles. So up to now, so we've already identified, we analyzed more than 135 genes. For instance, this included 79 genes from the 20 research group in 16 other institutions in China. So for example, there's just one example published by the Zifeng Guo's group from the Institute of Botany in Chinese Academy of Science. So they identified some genes, GELP96 genes. So this genes regulate the spike fertility, and so they also identified the mutants which validate the gene functions. So in the near future, there will be more and more reports to be studies to be published with this mutagenesis, HTX mutagenesis preparations. And finally, I would like to give a summary. So in this talk, so firstly, I would want to mention we generated a reference guided teaching platforms. So right now we use the amplicon-seq approach to identify the mutants. It will usually take 3 to 4 months to get the seeds once you provided the genes for analysis. However, anyway, so these resources would be very important resource for the forward and reverse studies in barley. So secondly, so I hope I already show you clearly. So if we combine the mutagenesis preparation together with the genomic tools, we are able to rapidly identify the genes, whatever is qualitative or quantitative traits. So in barleys, if my talk is not clear enough, you are welcome to look at our papers, so which is published in 2024. So in plant communications. And finally, I would like to appreciate all the members in my groups. So Chongchong Jiang is basically working on this mutagenesis preparation, and Miao Miao Guangqi also performs the work in the field and the bioinformatic analysis. Wen Xue Wu is responsible for, she is working on the S2 locus, and Sun Man, so she is working on the MF locus, and also other members who contribute to this work from my group. Of course, so the work can't be done if without the help from the collaborators. So I would like to appreciate Martin Masher and Niels for the contribution to generate the reference genomes for the HTX, and the Manuel and George for the genome annotation, and Chunmin Liu and Xuefeng Yao, so they help us to analyze the mutant frequencies. So the FS use the traditional approach. Of course, our work gets help from the so my colleagues from other groups. So in our institute, and also some students from the Professor Zhongyun Feng's group, so work in this project. Of course, finally, I would like to thank all the funding agency for the kind to provide the resources to conduct this work. And finally, thank you for your attention.
Thank you, Ping. I think we're going to continue directly with the talk from your student, and then we will take the question for the both talk at the end.
Okay. Yeah.
Hello, everyone. Thanks the International Barley Hub give me the opportunity to share my work here. My name is Guangqi Gao. My supervisor is Professor Ping Yang. The title of my presentation is Genomic Selection and Genetic Improvement of Chinese Barley Varieties. The content of my presentation include background, significance, progress, summary, and next plans. Firstly, the background. Cultivated barley is the fourth major cereal crop after maize, rice, and wheat in per se, and it is a self-pollinating diploid crop and can be served as a model crop for genetic research in treatises. It can be conducted genetic transformation and the construction of mutant libraries. And there are abundant reference genome resources of barley, for example, the Morex and the pangenome version 1, version 2. And there are abundant germplasm resources, include relative species, wild, semi-wild, landrace, and cultivar. Barley can be classified into hulled and naked barley based on whether the mature grain are adhered to the hulls. The naked barley is known as qingke in Tibet, and based on the row type, barley can be classified into 2-rolled and 6-rolled barley. In production, about 75% of barley is used for forage, and about 20% of barley is used for brewing, and about 5% of barley is used for making healthy food. In China, barley breeding can be classified into 3 stages. Stage 1 from 1950s to 1970s. The goal of this stage is to address the food security, therefore increasing yield, enhancing disease resistance, and improving stress tolerance were the breeding objectives for barley. Stage 2 from 1980s to the end of 20th century. In this stage, with the rise of beer industry, there was extensive utilization of exotic lines for breeding malting barley. And in stage 3, from the 21st century to the present. In this stage, driven by healthy conscious consumption and the rapid growth of livestock farming, breeding efforts were focused on addressing the quality requirements. So far, with the rapid development of China's livestock industry, the prosperity of beer industry, and the emergence of barley health food, the demand for barley in China has been increasing year by year. But compared to other barley producing countries, there is a significant potential for increasing the yield of barley in China. And China is the world's largest barley importing country. So there is an urgent need to breeding high yielding and high quality barley varieties in China. The significance of this study. This study is based on population genomics, aiming to uncover the changes of barley agronomic traits driven by breeders, elucidate the genomic selection basis behind these trait changes, and analyze the genomic structure of barley varieties. By using GWAS and selective sweep, we aim to identify the selected candidate genes and the genomic regions. And the candidate genes will be validated and cloned using reverse genetics experimental methods such as mutants validation and gene editing, thereby providing genetic resources and theoretical support for barley genetic improvement. The progress of this study. In Chinese barley breeding, these two books document nearly all Chinese barley varieties that promoted and planted in China. The first book records 309 barley varieties in China from 1950 to 1985. With 294 samples were used in our study. And the second book records 202 samples from 1986 to 2015, with 124 materials were used in our study. Additionally, we also our materials also include 87 new varieties released since 2016 and 27 exotic lands. Totally 532 historical materials were used in our study, and we conducted 6 experiments over 2 years and 3 locations for phenotyping, which include 10 agronomic traits, 1 BAYMV resistant trait, and 2 quality traits. The 10 agronomic traits are mostly normal distribution, indicating that they are complex quantitative traits controlled by multiple genes. How these genes changed during the barley breeding in China? Based on the released years, we divided our materials into 3 groups and the exotic lines for phenotype comparison. As the breeding progressed, the hidden data advanced, plant head reduced, the spike length, seed number per row, 1,000 seed weight, seed length, and seed wise all increased. So the genomic selection and the candidate gene variation behind the phenotypic variation require further analysis. Then we selected the samples for whole genome resequencing. Firstly, we conducted the genetic diversity analysis on these 532 samples by using the Barplex version 1 genotyping method. This method was published on Plant Communications this year. After excluding the redundant samples and heterozygous samples, a total of 384 accessions were selected for whole genome resequencing. The mean depth is about 6. The max depth is about 11, and the average depth is about 7. After filtering, about 13 million SNPs were obtained. In the genetic diversity analysis, we can see the C1940 and 50 varieties had the lowest nucleotide diversity. After 1960, hybrid breeding with exotic lines led to an increase in nucleotide diversity. In the differentiation index analysis, the varieties released after 1990 showed the lowest FST with exotic lines. indicating that a close genetic relationship between the two groups. From LD decay, the C1940 and 50 varieties exhibited the lowest LD decay, indicating a low genetic diversity in this group. And the varieties released after 1960 showed a faster LD decay, indicating that hybrid breeding using exotic lines has increased the genetic diversity of Chinese barley varieties. In PCA analysis, the Chinese barley varieties can be classified into 3 clades. Clade 1 mainly consists of Chinese early varieties. Clade 2 mainly consists of Qingke varieties. And clade 3 mainly consists of modern barley varieties, which cluster together with exotic lines. The phylogenetic tree also showed the similar results. And in the structure analysis, when k equal to 3, the exotic lines mainly exhibit the green component, but the Chinese early varieties showed the red component. As the breeding progressed, the red component in Chinese early varieties gradually be discarded, and the green component gradually becomes the predominant component in Chinese barley varieties. So these results showed that modern barley varieties bred in China exhibit a high similarity to exotic lines. Then we conducted the PCA analysis of Chinese historical varieties on a global scale. The Chinese early varieties primarily concentrated in East Asia, and the Qingke varieties primarily concentrated in Central Asia, and the modern barley varieties primarily concentrated in here. This result indicating that the modern barley varieties bred in China already possess the genomic framework of exotic lines such as the Europe barley varieties. Then XPCLR FFT Pareto analysis were conducted, and the top 5% and the top 1% were selected as breeding selection singles. When top 5% as threshold, 3,000 to 5,000 selected loci were identified. And when top 1% as the threshold, there are 600 to 1,000 selected loci were identified. The two examples of WOS1 and NAD showed the reliability of our genotype data. That WOS1 can be detected in 6-row barley versus 2-row barley, and the NAD loci were detected in hulled barley versus naked barley. Then we divided our genotype data into 5 sets. The first is all samples and only hulled barley, only naked barley, only 6-row barley, and only 2 barley. And GWAS was conducted on these 13 phenotypic data sets from 6 experiments over 2 years at 3 locations. From GWAS, a large number of associated loci were identified, and here are the examples of WOS1 and NAD. From the 2 examples, the 2 examples showed the reliability of our GWAS workflow. Then about hidden data, here are some examples. We identified the VNH1, HVFT2, PPDH1, the new QTL on chromosome 7. For plant hit, we detected the BRI1, VNH1, a new QTL on chromosome 1, and a new QTL on chromosome 3. For spike length, we detected a new significant QTL on chromosome 2. Then the summary. Through phenotyping on 532 historical materials in China, we reviewed the trend of changes in agronomic traits during the breeding process. Through the genetic diversity analysis of 384 historical barley materials, we found that hybrid breeding with exotic lines had increased the diversity, and modern barley varieties bred in China had already possessed the genomic framework of exotic lines. From GWAS and selective sweep, we have identified a lot of new associated loci. Next, the haplotype analysis, mutant validation, gene editing, and reverse genetics validation were used to clone the candidate genes that applied to genetic improvement in China barley breeding. And analyzing the founder genotypes combined with the phenotype variations to decipher the history of barley breeding transitions. Thanks the Barley Genetic Resources Group of CAAS and my supervisor, Professor Ping Yang, and thanks the Domestication Genomics Group of IPK. And the germplasm resources are from National Crop Gene Bank of China and IPK Gene Bank. Thanks for attention.
Thank you for your talk. So we're going to ask the question. Some questions are indicated in the chat box. Maybe for people that want to ask a question themselves, I will let them to first ask a question. Otherwise, I will read it. The first question that I can see on the chat is from Roby. Do you want to ask it directly, Roby?
Sure, if I can find it. I'm now scrolling up past the top. What did I ask?
Do you have any the clue about the history behind HTX?
Oh yeah, you showed that HTX came from somewhere between Ethiopia and European germplasm. Do you have any idea about the history of HTX, where it actually did come from? So when did it arrive in China? Why did it become important?
So this acquisition was collected from Harbin, so the northeast part of China. So this acquisition was available should be more than 40 years ago, more than 40 years in China. I speculate this might be spread to China through Russia, but however, we don't know the path, how it come to China. We don't know it, but we know it was in the Chinese collections for more than 40 years.
Okay. It'd be a nice story. Thank you. Ping, I have to go. I've got to meet a visitor now, so please enjoy the rest of the questions.
Yeah, thanks.
Okay, the next question is from Avinash. I can leave to the person a few seconds if you want to ask himself. Okay, I will ask it. Is there work on TSTOL1 gene and BNI1 gene, BNI gene in barley?
Which gene once again?
TSTOL1 gene and BNI gene in barley. The question is, so far, do you know if there is any work on this in your program or?
Ping, before I go, Ping, could I ask you to take the presentation off the screen so we can see everybody's faces?
Because sure. I stop it. Yeah.
Yeah, it's just that we can't see people put their hand up.
Better, because I'm not familiar with this software.
No, that's fine.
I use it for the first time. Yeah.
There we go. Now we can see everybody. Perfect. I'll see you later.
sir, this is related with the absorption of phosphate and nitrogen. So this kind of work is doing in wheat. So have you any information such kind of work in barley also?
I did not get your point for the first sentence.
These are the two genes that is related with the fertilizer use efficiency. Pistol one is related with phosphate and BNI related with nitrogen. So this kind of work is started in wheat. Do you know any work, such kind of work in barley?
I have no clue. Yeah, I don't know.
Okay, sir. Thank you.
You're welcome.
The other question, I don't know who is this person, so I will ask it for that person. Is HTX a cleistogamous barley or non-cleistogamy? Basically, is the floret open naturally or not in HTX? What about the
I
lodicule?
think it's flower. It's closed flowering, not open flowering.
Okay, okay. The other question is, do you know what is the yield average in ton by hectare of HTX? And what is the content, the malt content, or the percentage of starch? You already mentioned in your presentation the percentage. So basically the question is, what is the yield in ton hectare for HTX?
So HTX is a landraces. So so if they grows in the grows and mix soil in waters, so it will fall down like the all the landraces. It falls down before maturities. So yield production is not high, but this land races show the very higher thousand grain weight, more than 55 grams per 1,000 grains. So I think this would be much lower. Yield production would be much lower than the modern varieties. Why we use this lines? We also considered we want to mimic the process to select it for barley breeding from the landraces to the varieties, to the modern cultivars. We want to know what kind of genes or what kind of genes have been modified from the landraces to the cultivars. So that's why we selected this line. Of course, this line also is virus susceptible. So we are also working on the virus susceptible resistance. So the barley yellow mosaic virus is resistance is mostly conferred by the recessive genes. So that means loss of function of host factor genes will confer resistance. So this line is susceptible. So if we test it for the resistance in the mutant, we might be able to identify some Lyme's resistance. So that would allow us to identify the genes behind. So that would be one of the reason to work on this material. Yeah, thanks.
Okay. I think there is someone who raised this question. Maybe the person can turn on his microphone and ask the question directly.
Can you hear me, Ping?
Yes.
Thank you. Nice presentations. I'm just wondering.
Hello, Jay.
Hi, how are you?
Yeah, I'm fine.
Yeah. No, I'm just wondering how easy for us from Europe to get access to the HTX material from China, and would you collaborate also with the commercial company, for instance?
I mean, this is a very important question we also think about a lot. So, because with the current regulation in China, would be very difficult to export the mutant to abroad. But however, we also have some collaborators from other countries, so they can tell us the genes they are working on. If the phenotype is not very complicated, we will be happy to identify the mutant and perform some experiment to validate the gene function or to analyze the gene function. So this is one options. Another options next year. So the Highland province will become open. So to the foreigner to the abroad. So I hope it seems there will be something regulation change. So in Highland province, so the island at the south part of China, so they will be open. So in next year, so the foreigner students go to Highlands, Iceland, they can travel with visa free. So there will be something different. So that means there will be some becomes easier for the material exchange. Yeah.
Okay, thank you.
Thanks.
There is another question in the chat. This is from Newport. Maybe that person can ask the question directly.
Can you hear me? Because my internet is not very well.
Yeah,
Yeah?
it's very good.
Oh, okay. Oh, well, I was wondering that this is for Guangxi. Like, I was thinking these 3 different locations where you did your field trials. Are they all in the same province or are they in different, like, are they all in China? I mean, maybe you mentioned it in your presentation, but I might have missed it.
Sorry?
The
I don't get.
the locations for your field trials, are they all in the same province or are they all in a different place, like different provinces of China, or?
Yes, we planted the materials in different provinces of China. For example, the Henan, Yunnan, and the Beijing province.
Okay, that's all I wanted to know. Thank you.
Okay, thank you.
I don't see other hands raised or other question. Maybe I will finish with a personal question I will have. Is the black color of your landrace that you're using, do you know the genetic basis that control this dark black color of the old plant and the grain, or it's remain unknown?
It's BRP1.
Okay.
So the black Nima one. So this genes was so this is a very well-known material. Not so the black traits is well-known. So I know several groups who are working on that. So last year, so there's one group. So from the students of Chengdao. So so he collaborated with Chengdao to publish this BRP genes in Plant Biotechnology Journal. So they identified the AVT1. This gene would be the one component to cause the black Lima phenotype. So this is a very wonderful work, I would say. So we also did some work, but not direct from my group, but we collaborated with Alessandro from the Italy, so the CREA, Genome Diversity Center, CREA. So we also found some genomic variation at these locals, and we believe so this locus is very complicated. There must be several copies genes, and I also believe there will be additional genes are responsible for the black phenotype. So it's likely it's not a single gene. There would be several genes work together to lead to the black Lima phenotype. Regarding to, because we also observed some different on the black color. So because so for some black genotype, so only the greens becomes black. So the other tissues are greens, but however, so the HTX will observe something different. We already have one mutant which shows something different. So the green is black, but the limas and also the leaf tissue, vegetative tissues are yellows. And this gene seems is not black BRP1, but or genes close to BRP1, but definitely there will be additional genes. So to regulate maybe the movement or translocation of the black, so the melanins from the green to other tissues. Yeah.
Interesting. Thank you very much. So there is no other question, so I will just thank Ping and Guanqi for the presentation today. It was very interesting, well done. So thank you to participate to the IBH this morning.
Yeah, thank you very much, Sebastian. Bye-bye.
Bye-bye.